Good afternoon, everyone. My name is Francisco Villaplana, and I am Associate Professor at KTS, the Royal Institute of Technology, and I am also a member of the Wallenberg Science Center and also of Research. And it is my pleasure to coordinate this series of webinars on the characterization of wood biopolymers. So we started before last year in 2021, where we had two webinars by Professor Miriam Cabell and Professor Beta13 that were dealing mainly with the characterization of wood biopolymers using mass spectrometry and liquid chromatography. And for this new year, we will continue in that pathway. So we will have now another block of two webinars where we will be looking more specifically at different techniques or approaches to measure the molecular weight of biopolymers. And it is my high pleasure today to introduce Professor Bjorn Christensen from the Norwegian University of Science and Technology. He's a professor at the Department of Biotechnology and Food Sciences, and he's also part of Novipol, which is the laboratory of the Norwegian Biopolymer Laboratory, where you are working on the characterization of mainly marine biopolymers, but I guess not only those ones. And you're developing, as I said, new concepts, new materials, enzyme technologies, and also very importantly, different experimental protocols to understand the molecular structure of these biopolymers. But specifically today, so Bjorn will be talking about multi-detector size exclusion chromatography, so basically as a tool to understand the behavior of polysaccharides in solution, and talking about size on molar mass distributions, and also very importantly the stiffness of the chain and chain extension. So again, it's a pleasure, Bjorn, that you accepted to be here today with us. And basically, you can start whenever you want. And for the audience, Bjorn was mentioning that if you want to maybe ask some questions on the way, you're very welcome to do it, maybe in some of the natural pauses of the presentation. So just write a question in the chat. I will be moderating it. And also, if you want, you can also raise your hand, and we can also give you the opportunity to ask the question in real life. Okay, so Bjorn, all to you.
Thank you, Francisco. First, I'd like to thank you for inviting me to give a lecture at this course or this lecture series. I'm very honored to be asked. The topic of my talk is something we are doing all the time here. We are using multi-detector SEC on polysaccharides in solution almost all the time to monitor the size, to see if it has been degraded, etc., etc., and sometimes to investigate new type of architectures and chain extensions and so on. So over the years, we have had all kinds of polysaccharides through our columns. Some are easier than others. So I'll show at the end, after going through some basics quite quickly, I'll show some examples that I hope you find relevant. Okay, so let's move on. Polysaccharides, the world of polysaccharides is known to maybe most of you, but to many people, even scientists, and they are not so well known. And sugars, you are probably well aware of how they may look like, but also when you dissolve a polysaccharide in solution, just like any other polymer, they may experience very different chain stiffness. And this can be measured by parameters like the persistent length, which you can obtain from these techniques. I'll show later how we do it. And for instance, the 1,6 linked dextran, this is a very flexible type of geometry between the sugars, so it has a low persistent length. Quite similar to many synthetic CC polymers like PVC or something like that. Then as we move away from 1,6 linkages and over to 1,4 and 1,3, we see they become much stiffer. They will have persistent lengths in the order 5 to 15 or even larger nanometer. And then there's a big jump to things like xanthan and schizophile, because they can adopt a double or triple strand, which are very stiff, almost rod-like. So all this we can see by these techniques by monitoring molecular weight, but a second, say, radius or intrinsic viscosity in addition. So this is the backdrop of my presentation. The size exclusion chromatography, which is the same as GPC, is a technique many of you may know. The column separates according to size, and the particles are simply cauliflower-like particles with pores in the size range where you want to do your separation. So that means you may have many columns sets in your lab, and sometimes you need to switch column to adapt to your material. So if you put on a dispersed sample, most biopolymers, most polysaccharides are dispersed, or that's the same as polydispersed. But some years ago, UPAC recommended scientists to use the term disperse, and what we have called monodisperse was recommended to be uniform. So I will use both. So you put on with different sizes, and they migrate at different rates to the column, and you at the end, at the detector, which is usually as minimum a concentration detector, you will get a chromatogram. However, if you calibrate your system, say you have standards, you will quickly note that different polysaccharides have different calibration curves. So, for instance, an alginate of 100,000 molecular weight will come at around 22 mil on this column, whereas a polyolane with 100,000 would come, you have to extrapolate, very late. And this is because polysaccharides have so different shapes and extensions in solution, as illustrated in this blue box. So the separation is not according to molecular weight, it's according to the volume of the molecule in solution. And many people buy dextran standards because they're easy to get, but dextran and also polyolane are quite non-representative for other polysaccharides, in fact. So everybody gets a surprise when they compare calibration with a standard with the SECMOLS data later. So therefore, calibration only if you have the same type of polymer that you're going to investigate, and they are very rare to get, in fact. So if you do a multi-detector SEC, you can avoid this calibration problem, and it's often referred to as an absolute method. And the system would then contain a concentration detector, and usually we use RI, meaning refractive index. Protein people or nucleic acid people can use UV also. Then we have a light scattering detector, which provides a Rayleigh factor for different angles. And optionally, we can attach a viscosity detector, which sometimes gives very useful information, which you cannot get otherwise. The scattering from a solution containing a polymer is well understood. All instruments have usually a cell, optical cell, onto which a laser shines a strong laser light, and you have a detection system consisting of photodiodes sitting at fixed angles. In the instrument we have this fixed angle. Some instruments can move the detector around. And what we measure in this experiment is the intensity of the scattered light for different angles theta relative to the incident beam I0, and with a geometric factor, and all this combined is called the Rayleigh factor. So you can say this is what we measure, and what we measure is related to the biopolymer in several ways. This standard equation looks a bit complicated, but this is the form that's most useful. So you see there are two parentheses here. To the right, you see it's a function of molecular weight and the concentration with some factor, but also it's a function of the angle. It's a function of the wavelength, which you know. It's a function of the radius, which I come back to. And again, the concentration also appears here. And finally, there is this K, which is called the optical constant, and it hides the wavelength, again, which you enter in the program. It contains the refractive index of your solvent. So if it's water, it's 1.33. And this little mysterious DNDC, which you always need. What is that? The DNDC, or the refractive index increment, it's related to how it's the optical contrast of the system. And that polysaccharides are often in the range around 0.15 in water. It can vary. It's very important to determine it because in some cases it can be very different. And why this letter mu, or mu in English? If you have a polyelectrolyte, which you often have, if you have charges, carboxyl or sulfate or sulfonate, you need to establish equilibrium because this interaction with ions influences the refractivity. So that will sometimes involve the dialysis of your material against a solvent before you measure DNDC in a separate experiment. Or you're lucky and find it in literature for your system. Many scientists forget this a little bit. I could add, for instance, that if you go to carrageenans, which are highly sulfated, this value is down to 0.0105. So it's much, much lower than many of the, says, alginate or xanthan or dextran. So this is very quickly the theory. If you do an experiment and use the software in the right way, you can observe somewhere in the menu your first result. And since you have a detector that takes the concentration, you will see the concentration profile. Here you see a broad peak, so this is a dispersed sample. There is a salt peak because of the buffer you're using. Also the negative peak has to do with the salt. If we plot all the detector outputs from low angle to high angle, you can see that it may be difficult to see in this plot, but if I look quite late in the peak, you may see that the scattering intensity is roughly the same, meaning the scattering is independent of the scattering angle. But if you move to the left in the chromatogram, I try to hit the peak here, you see that the intensity gets stronger and stronger and stronger as we move to lower angles. So large molecules, the scattering is dependent on the scattering angle. And this dependency will give us radius of gyration in addition to molecular weight for large molecules. Here I have added a simplification of the formula, which is very useful to understand sec-moles. If I just consider a very low angle and low concentration to simplify, you will see that the scattering intensity is a product of C and M. This confuses many because in other detectors, it's always a matter of C. But if you have a sample with a certain concentration, with a certain scattering, and you degrade it with an enzyme or chemically to a lower molecular weight, but the same concentration, it scatters less. Simple as that. So how do we deal with the data? Of course, much is done by the software, so you don't see all this, but we need in some way to get the concentration in gram per mil, not in molar, but in gram per mil. And you have a detector output, some voltage for each slice. We divide the chromatogram up in maybe a couple of thousand slices. And we subtract the baseline voltage due to the scattering from the solvent. And the concentration is some voltage converted to refractive index by this ratio, converted to concentration by this ratio. And if you look upon it a little bit, we'll find that this parenthesis is an instrument constant that is known, or you calibrate your detector every now and then. Usually it stays constant over years. And secondly, this ratio between concentration and refractive index by reorganization becomes this DNDC. There are several reasons for having that as a differential. I will not go into that, but it's really the ratio between concentration and refractive index. So you see, DNDC is needed not only for light scattering. It was also a part of the K constant, but it's also needed to get the concentration. And this is a bit of a bonus, because in light scattering, you would see that the molecular weight would actually be proportional, inversely proportional to the square root of DNDC. So wrong DNDC, quite wrong MW. In SEC-LS, it kind of cancels. So it gets only the power of 1. So 10% wrong DNDC, 10% wrong MW. So with all of this data in the computer memory, the softwares will usually do the job for you. And here is a print of an Astra page we use a lot. It's an intermediate type of investigation. Here, you see the blue is the concentration profile. So it's a dispersed sample. The red is a 90-degree scattering, and you see it's shifted to the left. And that is simply because small molecules coming here scatter very little. Big molecules scatter a lot, even if there is not so much of it. So this is only when the sample is uniform or monodispersed do they coincide. So the software will stop at each slice. This is happened to be slice number 1519. And we calculate the factor K times C. C has been obtained from the RI detector. So here is the value. We have all the angles, and we extrapolate to zero angle, giving us the molecular weight at this point, 4.79 times 10 to the 5. And from the slope, we get the radius of gyration. So here are the formula. It's just using the basic formula to get what we need. So the intercept will give us this. The A2, the second virial coefficient, is something you have to tell the software what to do. But we'll see later that this term usually can be ignored, not always, but very often. So we get the molecular weight at this point from the intercept, and the slope by recalculation, we get the radius. So the software will do this simultaneously for all 2,000 or 3,000 slices, and you get then, as we will see, the radius and the molecular weight. But before we continue, what is the radius of gyration really? This is a bit confusing term because gyration would makes at least me to think about a bicycle wheel or something. But we are not studying rotation or movement like this. The definition of radius of gyration, RG squared, it's simply the distribution of mass around the center of mass. So this is the definition. Of course, we do not know where the center of mass is, and we do not know all the distances, but light scattering will provide it anyway, without assuming anything. This is very important. Light scattering gives radius without assuming the shape. Because in the mathematics of the light scattering, the treatment of the data lies this expression. So we get it out. So in other words, RG expresses the distribution of mass around the center of mass. So if the molecule is very expanded, RG is large. If it's very contracted or compact, it's small. And if you have a protein which denaturates, you can see that RG changes. For taking an example. So this was RG. I said the role of A2 is usually small, and that's because in sec-mols we use very, very low concentrations. So an example from our lab, even the peak is this low. And when you insert that, you find that it doesn't matter so much. So as a result, usually 2A2C is much smaller than 1/m. So you can ignore it, and you can set A2 to 0 in many cases. Only in special cases when C is large, you may see an effect. And you must stay a bit awake when you do a new polymer for the first time. So this is the exception. So there are many ways to present your data. It all lies in the computer memory now. So you can ask it for almost anything. And this is a typical way I usually present it in publications and so on. I show the concentration profile. I may include the 90-degree detector angle, and then we plot the calculated molecular weight across the profile. So each data point is a calculation of the molecular weight, as I showed in the previous slide. And of course, at the flanks here, you get noise because the weak signals difficult to define from the baseline. Here, at the low molecular weight tail, you see that the scattering gets very low. So it gets a bit noisy, and sometimes you have to extrapolate here to count everything. So simply, I have used this from using the software has used the basic equation to get the data. So in the computer memory lies now a big table, a big data matrix of the slices from 1 to maybe 2000. Each has a volume in milliliter. Each has a concentration calculated from the RI detector in gram per mil. We have now found the molecular weight at each point, and we have the radius at each point, hopefully. And this is the same figure as before. Having all these data, you can use the formula for number and weight average and calculate that. Of course, this is reported by the software directly. We get the radius data as in the right column, and a plot of radius across the chromatography file, the chromatogram, like same as above. You see that it also falls because molecules get shorter and shorter. It goes in this case from about a little more than 1 million to about a little less than 100,000, and the radius goes from about 120 for the big one. And we can monitor it down to about 20 nanometer in this case. Then it gets very noisy, despite the fact that we have scattering. So here is the scattering. There's a little bit of scattering. This is a problem in light scattering, that we cannot see a radius when the molecules are small, because this slope that we used to determine RG, it becomes smaller and smaller, and of course the data gets noisy. So the only way to get more data here is to go to lower wavelengths. Then you get more data, but light scattering is a bit restricted. What you can in practice do, you cannot go to 200 nanometer because everything starts to absorb. You may get fluorescence problems, absorption problems, and so on. And but you can continue all the way to the X-ray range. Then it's X-ray scattering. Then you get these nice data. But then X-ray scattering is not a small instrument in the lab. Many of you may know what it takes to do X-ray scattering, but it's the same type of reasoning behind it. Okay, so we have obtained the average molecular weights. We can plot the data in many ways. I included this also to show you this limitation regarding RG. The blue is an alginate sample. Here is radius again, down to about 20 nanometer. And here are lots of PEO standards. And you see, even if we have big peaks, it's only noise. Maybe for this sample, we have a well-defined, and here well-defined RG. So that is the reason for radius data below approximately 25 nanometer. Due to this limit, we set to roughly lambda 20th. So lambda is your laser wavelength. So you can't do anything about that. You can plot the distributions, which is sometimes very useful if you want to judge how much of your sample is above 100,000. For instance, in pharmaceuticals or even in drugs, you need to define the molecular weight range, and you may have to have your sample free of some smaller high ranges. And it's very useful to read this off. Then we can move on. In Astra, you find something called a conformation plot. And that is a plot of the radius as a function of molar mass. So it's the same data as you saw before. The molar masses was this one line crossing, and the radius was the other one. And you see they fall on a very nice line like this. And there is a theory, you fit that, and you can look up any polymer textbook. You will find that a solid sphere will scale to the power of 1/3. And this is simple geometric considerations. It will be the same for a glass bead. For a rigid rod, it's proportional. For a random coil, it has an exponent of 0.5 to 0.6, 0.5 in a so-called theta solvent, and then 0.6 in a good solvent. So if you have good data for a good span of molecular weights, you get this relation very nice. And in this case, I think the slope is 0.57. It's a random coil in a good solvent, and it happened to be a Kaitosan at pH 4.5. So a clean sample, having a broad distribution, good data, you get this straight out. So, but as I said, sometimes you do not get radius data. Then you can add a viscosity detector to your system. It's not cheap, it's not inexpensive, but if you have it, we have it, it tends to give useful data because it will give the intrinsic viscosity for each slice. In addition, this is and the good thing is you can measure it all the way across your peak. There is no limitation like in RG. I will not say much about how this detector works, except that inside the box, the flow is split in two. You measure the pressure differences 2 places, and you have a hold up, a little column or a dead volume here. So you measure at one place, you measure the sample, and the other is the solvent. And getting the specific viscosity and the intrinsic viscosity is just based on the pressure differences. So this we sometimes do. And I will show some data later, so I skip this. But in principle, you can get for a single experiment, you get molar mass distribution across the peak. You do not always get the radius if the molecules are small. We have to stop here. It was very noisy, but here you see the intrinsic viscosity across the whole peak. So you get simply more data. So in the second part, before I continue on that, do any of you have a question to the first part? So I can spend a few minutes clarifying concepts, if you like. You can grab your phone or send a chat. Okay, a little coffee for me.
I can actually read. So Gregory has a question. So he's asking, how is the polymer, for example, pullulans conformation in a solution depending on the solvent, assuming 100% solubility? So how would it affect the molar mass determination?
That's a good question. I am trying to think if one of the examples coming afterwards may change that. I don't have pullulan, but if you have pullulan, and which water is a good solvent for that, the indicator for the quality of the solvent is the A2 value. So if you do a conventional light scattering experiment with many concentrations, you will get the A2. And typically that will be positive, indicating a good solvent. If you add a little bit of alcohol, I've never done this, but I can imagine you do that, the solvent will go from good to via theta to maybe a poor solvent, and in the end will precipitate. So normally the molecule contracts as you go from a good solvent to a less good solvent. Pullulan has no charges, so we always get the same result in all buffers, more or less. But Kaitosan or alginate or xanthan, everything with charges is very dependent on salt. So the molecular weight will be the same unless they aggregate. but we can see a difference in radius or intrinsic viscosity at two different ionic strengths. So if you set up your system with two different ionic strengths, say 10 millimolar and 100 millimolar, you will see a difference in Rg for almost all polyelectrolytes. Normally, we keep salt a little high to avoid and standardize it. But if you like, you can see this shrinking and expansion. If the chain is a bit flexible and has charges, then you see big effects. If you have a neutral, very few people have actually done this by sec-moles. It's a bit of a hassle to change the solvent. You have to equilibrate and wait overnight and so on, but it certainly is possible. I don't know if that answered your question. Yeah, and now I also see okay, that was good, thanks. So let's move on to some examples I have collected over the years. Most of them have been published. So we work a lot on alginates. They are polysaccharides from seaweeds or some bacteria. The question I wanted to address here is, does the chemistry influence the expansion of the molecule? Because it has been a little hot area. So here is a chain with only M. M means dimanuronic acid. This is an intermediate in the biosynthesis, but we can make it in bacteria by knocking out this step, the epimerization. This can be done. This is done in the plant. It's done by bacteria, but we can knock it out and study this. But we can also express the enzyme and do this in vitro. And this sugar is called L-glucuronic acid. And the conversion from M to G leads sugar chain to flip from 4C1 to 1C4, and 2 Gs form a kind of a cavity. This is a basis for calcium binding, but that's not what I'm going to discuss. We get all kind of block structures, and it's been written in literature for 40 years that these G blocks are stiffer than the M blocks and the MG. We had the opportunity to get these extreme materials. We got this polymanuronic acid. We have this almost exclusively G, and we also have one with MG, because there's one enzyme that makes that. And yeah, I'll skip this. We did conventional sec-moles for many different samples with the same type of chemistry. So I had the MMM, the MG, and GGG. Did the conventional study. First results looks just as we would expect. For the radius data, we have a normal plot, and here I overlaid it on polyulan just to illustrate that. Alginate is pushed very much up. It's much more expanded than polyulan. Such data can be analyzed. This you have to do by exporting the data into some other program or Excel. And for the low molecular weight range, you can use the worm-like chain model, fitting the results and finding this Q, this persistent length. What we found here was that it more or less in this range is about 15 nanometer for all alginates at this ionic strength. So in fact, this is the why this table I showed earlier shows 15 nanometer here. And we get for polyulan 1.3, etc. So this is how you do it. This is one way to do it. I will move on to Kaitosan. So if you look at Kaitosan and compare it to alginate for a minute, you will see that the basic geometry of the chain, at least the manuranan, is very similar. The sugaring are the same, except that, well, here is an axial hydroxyl and a carboxyl. Here is a amino, and it's equatorial. But there's one charge per sugar, so distance is roughly the same. And for simple polymer thinking might indicate, well, they will give the same chain extension under equal conditions. But we consistently find that the Kaitosans at the same ionic strength and fully charged is always more contracted than alginate for the same molecular weight, but of course stiffer than polyethylene. So just overlaying files. So you can take files from different experiments and overlay and see if polymers are different or similar. So you can see that Kaitosan is for some reason more compact than alginate. So they're a little more flexible, but we found that the flexibility is not so very much dependent on the acetylation degree. So this type of experiment, which you will accumulate over the years, they give a lot of information. We did another type of, in this case, we collected not only radius, but we did the same with intrinsic viscosity. I have not shown the raw data, but having molecular weights and having intrinsic viscosities, there is a theory called the worm-like chain for intrinsic viscosity. It's called the Bjorn plot, and we recalculate the data. So I calculate the combined parameter m squared over intrinsic viscosity to the power of 1/3, plot as a function of the square root. So a couple of new columns in the spreadsheet, plot them, they will be linear, and you read out intercept and slope, and you can get the persistent length. Here we compare the persistent length from RG data for different acetylation degrees. It seemed to increase a little bit from 4 to 7 by this method. Using intrinsic viscosity data and Bjorn plot, we get slightly different data. So take care if you calculate such a value and compare it to literature. Maybe it was not done in exactly the same way. So small differences can be hard to detect. Here you see all the data seem to lie on top of each other. This is a bit of a coincidence because the mass per unit length, of course, changes when you change the acetylation degree. Okay, I can move on. Many years ago, we studied Kaitosans in a different context. We did degradation of Kaitosan using nitrous acid. This is a quite old and classic technique, which will break glycosidic bonds stoichiometrically. So you add as much nitrous acid as you like or you need, and it will break this linkage and reorganize this sugar to another sugar, which is called 2,5-anhydro D-mannose. And what is special about that? It has this aldehyde as a pendant group. It's not part of the ring. It cannot easily form an hemiacetal. So this is very reactive. And that was the purpose also. One of the purposes to utilize that. But aldehydes like to react with amines. So the student, the PhD student at that time, Christopher Tømmerås, he noted that you didn't always get only shorter chains. Because what he did, he added sodium cyanoborohydride. Because if you get a shift base, it will be reduced and permanent. And now you get a branching. So did we get branching? Yeah, we can see this by NMR, even especially at high degrees of branching. But can we use secmols to see the same thing? So the idea is that when you get branches, you get more mass into the molecule, but it will not become more expanded. And here are 4 sets of data for different degrees of degradation. So here I had degraded to dp100, dp40, 20, and all the way to 10. And you see before and after. And you see especially when we degrade a lot, we get a lot of self-branching. And the whole curve moves and also becomes less steep. It loses its random coil type and it's on the way from random coil to something more sphere-like. Not much, but enough to detect. And you see it happen systematically. And we can calculate also the change in molecular weight from which we can calculate the degree of branching. So a very simple experiment comparing degraded and non-degraded Kaitosan, showing that it will self-branch. Okay, I moved to completely the opposite end of the scale. If you remember early in my talk, I had this table of persistent lengths. And at the bottom of that table was xanthan with a persistent length of about 120 nanometer. So it's very rod-like. This is one of the most studied polysaccharides in physical biochemistry, and big contributions from Japan, the Osaka lab, who did a lot of fundamental work. Much controversy over the years. But it's clear today that xanthan is double-stranded. It probably comes out of the bacterium. It's a bacterial polysaccharide as a double strand. We don't know the details here. A very good question for a good biochemist. But we can isolate xanthan, and xanthan is a commercial polysaccharide. It's a super high molecular weight, and it's very stiff. It's a fantastic viscosifier, and it's easy to produce, and it's inexpensive, in fact. So here on the left, you can see a published AFM picture from a British lab showing even the helical tendency of the double strand. So it's a double helix. 20 years earlier, we could use EM to see it was rigid, and we could sometimes see the splitting in two chains. So no doubt it was double-stranded. And then the question is, can we do this with secmols? Can we get the same information? Do we confirm or do we disconfirm literature? So we took a xanthan sample and we actually sonicated it. Sonication means ultrasound treatment with a very strong, very high energy, 300 watts per 100 mil. So it's a very powerful method. And today you can also use other methods to degrade mechanically. And they tend to break mechanically, so that just like a pencil, I will not break it. I think I would be able to break it here, but I wouldn't probably not be able to break the half of it easily. And we see the same thing with the sonication. It degrades quickly to begin with, but it flattens out. But here we made 3 samples, and you see a red chromatogram. So it degraded a little bit, blue degraded longer, and the green degraded even longer. But after that, it didn't move so much. And you see that the molecular weight curves for 3 different experiments, they overlay nicely. And this goes from about 1 million to about 100,000, in fact. So we degraded in this range. Is it double-stranded or not? We need more data. So actually, here are the weight average molecular weights, and you can also see reproducibility. When we do it again later, you never get the same value. We measured intrinsic viscosity with a viscometric detector. It's called ViscoStar, and we also did it offline. In fact, we later completed this data, but I found an old file. I'm sorry for not finding the one with all the data. So it's good to have this confirmed. We compared our data. So I made a plot of intrinsic viscosity versus molecular weight, and I plotted this with published data. The blue data are, I have to remove the picture of you guys to see my own slide. The blue data are published data from the Sato or the Nourishu group in Osaka in 1984, carefully degrading and analyzing NW and intrinsic viscosity. And here are our samples. They are quite close. They don't cover this wide interval. I couldn't get further down. I could have started with something bigger. But all in all, our data lie well on this line. So it confirms the data from the literature in this respect. Yeah, here is the double strand, see how we perceive xanthan today, the cellulose backbone with the side chains. I forgot to talk about the chemistry, but you will see here it's a cellulose-like backbone and with a side chain, making it water soluble and stabilizing it so it will not form a cellulose-like crystal. It is water soluble. Okay, so instead we could look again on our files. I took the different injections, plotted the radius data against molecular weight, and I overlaid the published data again. And you see also not only the average, but also I covered the same data as the literature. So I was very happy with that. It's good to have different molecular weight ranges, as you can see here, because every one of these, maybe the green one covers a sufficient large range. The others hardly do, and you see some problems at high molecular weight. This is due to weak signals, but also due to shear effects. It's very high shear in the system, especially for intrinsic viscosity. This is radius, so it's mostly due to weak signals at the flanks of the chromatogram. So it's good to have a couple and add them to see the trends. Here you see the Sato data. I just took that from literature. This is the rigid rod theoretical behavior. Slope 1, you see in low molecular weight, xanthan is very much rod-like. But as the molecular weight gets 1 million and 10 million, it starts to converge and approach a random coil. This is a phenomenon which is very important. So imagine you take a random coil like dextran and cut it up in pieces. In the end, the small pieces are like nails, so they are rod-like. So if you want to say a polysaccharide is a random coil or a rod or whatever, you must consider which molecular weight range you're in. So normally, all polysaccharides, all polymers, when the molecular weight is sufficient high, are random coils. But in the range you are operating normally, you may see rod-like behavior in the case of xanthan and random coils. Alginate, Kaitosan, and they are random coils in this range. And there's a theory, so you can fit the data as I showed earlier and get the Q value of 120. Fantastic publications from this group. Professor Sato, which will soon retire, told me it took him as a PhD student 1 year to make the samples before he started to measure. Today you can inject the next day. Okay. So I will leave xanthan and move to something completely different. We have worked for many years with Beta13 Glucans from fungi. This is an industry type of project. Fragments of the cell wall of Saccharomyces have interesting biological effects and were in fact developed towards a drug or a medical device. Other labs have done the same, and there's a lot of things going on with Beta13 Glucans, how they interact with the immune system through receptors and so on. And this immediately raises questions about what does the chain length mean, what does branching mean, because there is a little branching, and above all, what does aggregates mean? Because these materials, even this sample with a low DP, about 100 number average, it is not a solution, it's a gel at 2%, it's turbid. And if you dilute and inject into aqueous sec moles, you get this blue thing, this black type of data. It's quite irreproducible, a lot of scattering, and molecular weight starts in 10 to the 8. So 100 million, which is not very much in agreement with reducing and assay, which you can do. We developed 2 different ways to get around this problem. Either to modify it by carboxymethylation, and we were of course inspired by carboxymethyl cellulose, which has been around for a long time. This chemistry is quite simple. you can carboxymethylate, and we did that. And then this red curve, by the way, was obtained at 80 degrees. Still, it aggregates. But you see, when we carboxymethylate to first 23%, this is a green one, it's nice curved, move to the right. When you do more, it moves to the left again. And we think that is just because of charge effect. It expands a little bit more. But you get very nice data, very nice molecular weights, from which we can determine MW, MN, and so on. And then it agrees quite well with the reducing end. We did not do radius here. They are a bit too short to give radius data. And we did not do intrinsic viscosity either. That may come later. However, we were aware that especially cellulose labs, they do a lot with DMAC lithium chloride. So maybe Professor Antti Potast will later talk much more about this on cellulose. But we tested Beta13 Glucan in DMAC lithium chloride, and it dissolved very nicely. And in fact, we did secmols, and you get very nice peaks. This is the different peaks are just different time points after dissolution. So it dissolves very slowly. You get a higher and higher peak, but the molecular weight is the same. So meaning this particle you start with has the same composition throughout. It's a freeze-dried material. Later, I know that labs have seen differential extraction in this way, but here you can take any curve and get the same dP. Interestingly, when you do a new solvent, you have to do DNDC again. And DNDC, in this case, in this solvent, is difficult. If you look up DNDC literature for cellulose, you find anything from 0.078 to 0.078 to 0.15. I have discussed this a lot with Antti Potast. We try to estimate it ourselves by looking at the recovery after sec, assuming everything comes out, then you can calculate it. But it's slow dissolution, but there is no fractionation, and everything is in good agreement with this technique and with this technique. So by this, we have a technique for Beta13 Glucans that we can use. It works very nice. What about conformation or shape or stiffness? Well, the radius of this material is too low for LS. And even if we wanted, we cannot attach a viscosity detector with DMAC. DMAC is quite viscous, and the pressures become too high, and the producer will not let us do it without losing the guarantee. So we dare not do that, unfortunately. Okay, now we're reaching just almost 1 hour. I have a few more examples. Some years ago, I had a collaboration with Stockholm University and Professor Göran Widmalm. He had investigated a range of lactic acid bacterial polysaccharides, and he wondered simply, what is the molecular weight of these materials? So he sent me a lot. I picked three of them, the THS, the K16, and ST1. And I had a PhD student, Marianne, and also another one, and they investigated this by secmols. I wanted to know the molecular weight, but also can we see differences or similarities in chain stiffness? Are they similar to other polysaccharides? For instance, the THS has a kind of hyaluronic acid type backbone. But there are substituents. Okay, so there's a charge. There is no charge here. It's neutral. Hyaluronan has a carboxyl here. It has glucnac here, but it's 1,3, 1,4 beta beta. So maybe this behaved a little bit like hyaluronic acid. K16, I found nothing with this backbone to compare with that I had. ST1 has a 1,6 linkage in the main chain, but otherwise 1,3 and 1,4. So maybe this 1,6 would make it more flexible. This is what I predicted, and be a little more Polylan-like than the other. And I was right. But we did the experiments, and again, data look like this. A, B, and C refer to molar mass, radius, and intrinsic viscosity. And you see the chromatogram, the concentration is the same. It's the same data, just different lines. You can see here that K16 and THS, they give nice data across the whole peak, both for molecular weight, for radius, even down to 20 nanometer, and fairly well for intrinsic viscosity. But the ST1 have an upturn here and an upturn here. Many people ask me, what is this? This is sometimes called anchoring, but to be frankly honest, we don't know what it is. It can disappear if you purify better. But these samples I got unpurified. So this can be some material in the tail here that scatters light a little more than the background. It doesn't have to be much, but light scattering is very sensitive. That means we have a processing problem with this data, as we will see in a minute. But otherwise, it worked fine. Molecular weights are high. ST1 starts almost at 10 million. K16 at about 2 million. THS, yeah, about the same. But they are not very viscous, even with these high molecular weights. And even that told me that these are probably quite flexible chains compared to things we know from before. So we did a Norman analysis. We plotted radius against molar mass, the so-called conformation plot. And I added also here polylan and hyaluronan and also alginate. It's very interesting to do these comparisons. It gives a lot of information. We saw, in fact, that especially ST1 is not far from polylan. K16 in the middle, the most expanded, but still much less expanded than hyaluronan and alginate. The intrinsic viscosity data are a little bit like this, but stretched out. This is another advantage, that differences in RG become larger when you do intrinsic viscosity, especially for random coils. There's a reason for that. I will not go in. It's expected. And you see data, alginate stands out more. THS and K16, they are still quite similar. And ST1 is not that far from polylan. So even before doing some calculations here, yeah, this is reasonable. ST1 is similar to polylan because it has the 1,6 linkage here, which is very flexible. There are 3 single bonds between the rings. So this is like a flexible joint compared to the other ones. Much less steric hindrance, much less hindered rotation. So with this raw data, yeah, the slope are in the random coil range for all of these. You see slopes are different. Slopes are similar, but the vertical shift or the horizontal shift is different. So we concluded, we fitted those data to the Benoit-Doty, the worm-like chain model, and we got persistent lengths 9 nanometer for THS and 10 for K16, and lower between 4.5 and 5 nanometer for the ST1. It was difficult to get data here at the low end. So qualitatively, it makes all sense. The MHS exponents, the Markov-Sakurada exponential equation, which applies to intrinsic viscosity, were also as expected for random coils. So you see very nicely here that similar slopes, but different shifts, and that is due to the fact that two coils may be random coils, but one can be more expanded than the other. That's one way. Then it moves up, but also the mass per unit length. There can be substituents, can be more heavy. That moves it to the right, which is equivalent to going down. So it's a little bit difficult to judge too much from the shift before you consider the chemistry. So the 7th example is something we did a couple of years ago. Pyridate oxidation, you may know as a standard technique in carbohydrate chemistry, a bit old-fashioned. Long ago, it was used to find out which sugars are linked how, because it will react with vicinal diols. So in hyaluronan, it's only between C2 and C3 in the glucuronic acid ring you can oxidize. The rest is resistant, and it opens up and you get a dialdehyde. You can do chemistry on this. This is very hot these days. But also this opening of a ring makes this structure very flexible. So you have something quite rigid, and suddenly you open up, you have a point where you would expect it to move freely. And overall, this reduces the chain expansion and it collapses back. It becomes more contracted, and you would expect the radius to go down. You would expect the intrinsic viscosity to go down. We have done the same thing with alginate. I will not show that, but it works also nicely, and also with Kaitosan. So here are data from an article in 2013. So we have here the different oxidation degrees. You will see that the chromatograms are not that different. It moves a little to the right, meaning that it degrades a little bit. The blue chromatogram being bigger is just because we injected more material. The profiles, the molecular weight profiles, are quite overlapping. The radius are maybe the same, but you see intrinsic viscosity tried to drop a little bit as we oxidize. And that's the first sign of a compaction. And it's easier to see with intrinsic viscosity than radius, because for such random coils, the intrinsic viscosity is proportional to radius cubed. So a twofold change in Rg is an eightfold change in intrinsic viscosity for a random coil. That's why this is so sensitive for this type of events. And we did a lot of calculations. Here we see the degradation a little bit lowering a molecular weight, but it doesn't matter because we get the data across the profile. So, and then we analyzed again with the models I've shown before, the worm-like chain model. So here is the plot. We see that the persistent length drops here from 16 to 8. This is very influenced by the salt, so we can mathematically correct, and we see it moves from around 8. This is very close to the published value for hyaluronan at this ionic strength, and it drops down, and with 80% oxidation, it's down to 2. So, but even here with 40%, you have between 2 and 3. And this is interesting because you still have a lot of hyaluronan chemistry, but you start to get dextran or pullulan type physical chemistry or chain stiffness. So this is a very different molecule in solution. Intrinsic viscosity shows the same thing. And here you see more clearly the big, much bigger drop. And we can then keep the molecular weight constant. We can choose to compare at the constant molecular weight, for instance, 500,000. You see there's almost a factor 3 difference in intrinsic viscosity. And we can calculate again the same thing. Here are the previous data. Here are the new data. And the fitting, I have not I fitted the low molecular weight region. I have ignored the bending here. And I get qualitatively a little lower values, but they always decrease, as you see here. 2 different ways to treat the data. I will not go into that. It's in the paper. But these are standard ways to treat such data in literature even today. So oxidation of a hyaluronic acid opens the glucuronic acid ring. The whole molecule becomes much more flexible and it compacts. And it gets a gradually lower persistent length. This is my final example. This is something we have started to work on the last couple of years. And I talked about this at EPNO in Nantes in October. We have taken the G block out of alginate. This is the block of alginate which reacts with calcium. And we have coupled it to another block of means of a linker as part of a bigger project. And we predicted that this block is rather stiff. It has 15 nanometer persistent length. So when it's so short, it would be a rod, basically. But here it should be very flexible, and it should be what is called a broken rod. So we decided to try to determine the molecular weight, and also to see if we could get differences in intrinsic viscosity. So we added a viscosity detector, and here you see first the chromatographic peak. The left peak is the coupled product. The right peak is residual uncoupled, because this coupling is not very fast, and we stopped it before it was fully coupled. And we see the molecular weight. There are two levels here, one level for the dye block and one level roughly for the unreacted blocks. And in fact, the molecular weights, they correspond exactly to what we should get. This is G10, 10 sugars connected this way. And then we did also G23, because that's about twice as long as this. And we did intrinsic viscosity. So this is the same profile as here, but this is the intrinsic viscosity. So you see it's about 10 milliliter per gram across here, and it's roughly 6 here. We did the weight average. So you see G10 is 7. The dye block is 10, but a twice as long chain is 15. So, and this ratio, because if we had a rod, and you double molecular weight, you should have a 3.5 times higher intrinsic viscosity, theoretically, if it's a rod. It's not entirely a rod, but this difference tells us that this is a broken rod. It has high flexibility here. The molecule will move between different configurations like this, and on average, it's less expanded than the fully stretched chain like this. So this was recently published in Polymer Chemistry last year in August. So you can look that up. So I think it's time to stop. And if I wanted to summarize everything, sorry, I will start this again. This multi-detector SEC is a powerful and convenient method that we use a lot. In fact, we have 2 systems up running, except for right now. One of the detectors, the old one, is a bit sick, but we'll try to revive it. It gives us smaller mass distributions, that and the averages, of course. This is what we normally want. If it's a new polymer, we can get radiuses down to about 20 nanometer, not below that. If we have the viscosity detector, we get the intrinsic viscosity distribution and the averages. This you get if you have the D and DC and A2 issues fixed. A lot will change if you change this value. You better know what it is and take time to find it. If you're not sure how to do it, contact me and I'll show you where it's published and explain. It's simply dialyzing and measuring in a refractometer. That's simple. Once we have all this data, we can analyze our data much more in detail, and we can get information about chain stiffness in combination with an appropriate model. And that can tell us if it's rod-like or if it's coil-like, and if it's coil-like, it can give us the persistent length. It can reveal special architectures. If some part of the population suddenly is branched, you will see that as a bending of your data. And that's an important point. Most of these challenges are for a homologous series. Same chemistry, just shorter or longer chain. But if the chemistry changes, it's different for short and long. If there are branches for the long and not for the short, or vice versa, it gets much more complicated, and you have to take much more care. We can work in other systems than aqueous. DMSO works also. It's not so easy. DMSO tend to take up water. We were not very good at, and the columns were horribly expensive. DMAC lithium chloride, many labs do this routinely. Antipotas lab, for instance, and it gives beautiful light scattering results, because DMAC lithium chloride also dissolves everything, so there is no particles unless you have very complicated systems. You can derivatize your polysaccharide to change the properties in DMAC. Watch out for degradation if you do that. Yet there are challenges, of course. One of them is ultra-high molar masses. There you have techniques like field flow fractionation that looks very good on paper, but I have not done this myself. You may have variable architectures, say pectin, for instance, or even some hemicelluloses, I think are difficult. And not all columns are suited for light scattering detection. We found that new generation of some old columns leak nanoparticles, not for hours, not for days, but for months or years, and give so noisy. So we had to throw away a couple of columns. And one, you must carefully check your column when you buy one. So, ladies and gentlemen, I hope I presented some data that were useful for you, and some considerations and techniques that you can use in your own work. And don't hesitate to contact me if you want to learn more. So with that, I thank you very much for your attention, and I'm ready to answer more questions if you like. Thank you.
Thank you very much, Bjorn, for your comprehensive lecture, first on the basics and the background on size exclusion, and then some examples. So yeah, the floor is open for questions. So don't be shy. You can either write it on the chat or take the microphone and ask directly.
So I'm happy to go back to a certain figure and look at it with you again. No problem.
Maybe I can break the ice. So maybe you can actually go to the figure where you show the conformation plots for, a different range of polysaccharides. I mean, it doesn't matter which one.
Let me see, one where you had an overlay like this, for instance.
For example, yeah, that's a good one. So basically, they all show probably, they all show that they are random coils, probably because the slopes are quite similar. My question is very general. So, I mean, the difference is, of course, in how flexible the chains are. Do you have any rule of thumb based on the chemistry? So is there any particular type of polysaccharide linkages or architecture, molecular architectures that will give different flexibilities of the chains?
Yeah, in fact, and the best way to show that is maybe go back to the
first slide
table I had in the beginning, because this table really like this.
Oh yeah, you have it.
So those who are very flexible, they have typically alpha or 1,6 linkages. So many people have dextran in the lab, easy to get. You will see always you get very low intrinsic viscosity, low radius compared to the other, like the one I showed. So that is a reason, because the 1,6 linkage is, I'm trying to make it, if you can see from the camera. Normally sugars have the oxygen here and a linkage and a ring. Usually they are so restricted. Mm-hmm. But even if each of them is a bit restricted and you try 3 of them, I don't know, maybe you have done some fixing on your house or try to make some object which is not well secured, and you have many of them, you get enormous effects. So 1,6 linkages, they are very diagnostic. They immediately move everything down. And polyulan, we have done that a lot. It has alpha 1,6 and 2 alpha 1,4. Mm. So it is stiffer than dextran. You can see about a doubling in persistent length, because only 1 out of 3 is 1,6. then I haven't searched actively for all intermediates, but for instance, serial beta-13, beta-14 glucan, they again from 1,3 to 1,4, you are in this range, 3 to 7. Kaitosan, only beta-14, carboxymethyl cellulose, for instance, reported about 12 to 16. So it seems 1,6 is most flexible. Then I don't have anything for 1,2. Then you have 1,3. Then you have 1,4.
Mm.
But if you have alpha beta, that influences also.
Yeah.
So alpha, I think, makes it more. Alpha 1,4 gives, of course, starch like helix. But if you find a good solvent, the problem is not all of them are soluble in the same solvent.
Yeah, that's true.
So pullulan goes for DMSO, amylose for DMSO. I don't think, but cellulose not.
No.
There is a study from a Japanese group with comparing cellulose to beta-14 to cellulose to curdlan, which is only beta-13 in DMAC, and there you see the same shift. I can get you the publication if you want to.
Yeah,
take
just
a look.
because it's nice also to show it. It's curiosity.
I'll try to make a note. So
Thanks.
curdlan versus cellulose. I can send it to you afterwards, and you're free to distribute.
That'd be nice. Thank you.
I use that as a student exercise for my student. Yeah. And otherwise, you have this big jump, about one order of magnitude, when you go from a single strand to a double or a triple.
Even a triple. Yeah.
So I don't know how many are double-stranded, but xanthan at least is. It's not so easy to find out if they are double-stranded as you might think. That's why xanthan was so debated through the 80s and 90s, that even people didn't talk together, only shouted on meetings. This is when I entered the field as a young scientist. A bit strange. Scleroglucan Schizophile, they are fungal water-soluble polysaccharides that come out as a triple strand. And you can really do a lot and keep the triple strand. You can modify the small side chains and so on. It's really a little rod. So basically, yeah, 1,6, 1,3, 1,4, and then the double or triple. Yes. So maybe that's a thumb rule.
Thank you.
Yep.
More questions to Bjorn? Doesn't.
Yeah. But anyway, like my final advice is always, when you study something new, include some other standards that work well in your system. It's so helpful. So, for instance, the pullulan, which is commercial, you can buy. Hyaluronan, easy to get, water soluble, alginates, Kaitosan. But be aware if you have charges. Charges demand salt. So things like pullulan, which is neutral, it's a bit easier. There are other also. Okay.
Perfect. Thank you so much, Bjorn, again for your talk, and thank you everyone for being here today. And I actually am gonna advertise the next one, which will be, and as I said, in a couple of weeks, and it will be by Antje Podcast that Bjorn has referred to a couple of times in the talk. So, and I think she will be discussing maybe more in detail, probably size exclusion of cellulose in DMAC system, and specifically the challenges for lignin, which is another nightmare to work with in size exclusion, especially with light scattering, because you have to do, probably she will talk about it, to put filters or change the wavelength of the laser. So, but hopefully Ante will talk about all of this. So thank you very much, and I'll see you hopefully in a couple of weeks.
I hope so. Bye-bye.
Bye.